Welcome, everybody. Dr. Jason Huse is a professor in the Department of Pathology and Translational Molecular Pathology at the University of Texas MD Anderson Cancer Center. He's a neuropathologist specializing on the diagnosis and molecular characterization of brain tumors. He also directs a research lab studying the molecular pathogenesis of malignant gliomas and brain metastasis, and he's going to teach us today about rare gliomas.
Hi, so I hope everybody can hear me. Hearing no complaints, I will proceed. Thanks so much for tuning in and for having me here. I've been tasked to talk about rare gliomas, which is really an ever-growing subset of tumors whose establishment is being greatly facilitated by recent large-scale molecular profiling efforts, as you'll see, and a lot of which has impacted the latest edition of the WHO classification system from 2021. So just launch right in. Oops, let me, okay, I have nothing to disclose. Here is the learning objectives slide. Okay, all right, so first, like I said, I've been tasked to talk about rare gliomas. So let me start by telling you what I'm actually not going to talk about. I'm not going to talk about things, I guess, that are common. So I'm largely going to leave out IDH-wildty GBM and IDH-mutant astrocytomas and IDH-mutant and 1p19q codeleted oligodendrogliomas. Those compose sort of the majority of the adult diffuse gliomas, and I think they're common enough to sort of be left out of a discussion like this. I'm also not going to be talking about pilocytic astrocytoma or pleomorphic xanthoastrocytoma, which are also sort of the more common pediatric type lesions that you see. And just for the interest of time, because I can't actually talk about everything, I'm going to leave out glioneuronal and neuronal tumors, sort of these neuroepithelial variants that are, somewhat aligned to some of the pediatric type diffuse gliomas I'll talk about, but just for the interest of time, I'm not going to be able to really get into them. Okay, so on the other hand, what am I going to talk about? I'm going to try to focus on pediatric type high-grade diffuse gliomas, pediatric type low-grade diffuse gliomas. And why I say pediatric type is that although these are classically associated with the pediatric population, they can drift into the adult population as well, particularly into young adults. And so it's important that you keep these in mind when you're evaluating diffuse gliomas, even in adults. I'm also going to talk about a few of the circumscribed astrocytic glioma variants. And this is all, our touchstone document for this will be the WHO 2021 classification, which is actually formally codified a number of these entities for the first time. Okay. All right, so I was told that I need to have a case presentation, so here it is. A 4-year-old male with a history of intractable seizures and a left-sided non-contrast enhancing temporal lobe mass. So microscopic examination reveals a heterogeneous histopathological picture. There are areas that have pretty pronounced oligodendroglial differentiation with these monomorphic round oval nuclei, paranuclear halos, but we also have more astrocytic areas with increased nuclear pleomorphism. Interestingly, this lesion stains very strongly and very diffusely within its tumor cell population for CD34, and molecular testing reveals an FGFR2, ctDNA, and A3 fusion on molecular testing. So by the end of this talk, you will be able to slam dunk know exactly what this is if you don't already. I guarantee. Okay, so this is obviously, I'm trying to start off a beautiful relationship with you guys. And so, but first thing I do is actually lie to you. I told you that we weren't going to talk about IDH-mutant astrocytomas, and here I am. The first thing I'm going to do is talk about IDH-mutant astrocytomas. But this, I would argue, is actually a rare variant within an otherwise common entity. So IDH-mutant gliomas in general were thought for a long time to be entirely supratentorial, and it turns out that wasn't entirely the case, that they can actually arise infratentorially as well, the posterior fossa, brainstem, and even the spinal cord. It's unusual, but they do. But what's interesting about these tumors is they don't actually molecularly look exactly the same as a supratentorial IDH-mutant astrocytoma. So as you know, supratentorial IDH-mutant astrocytomas, they commonly feature the IDH1 R132H mutation to such an extent that you can actually use an antibody to IDH1R32H as a screening tool to detect these. They also predominate with ATRX mutations, and once again, an immunostain detecting loss of function for ATRX is a good way to actually screen for these. This actually is not really the case for infratentorial IDH-mutant astrocytoma, which actually there are two other mutations that actually are more common than IDH1R32H in terms of IDH1. ATRX is actually more often retained than lost, and there are actually differences in MGMT promoter methylation as well. So what this means pathogenically is not entirely clear, but simply from a pragmatic standpoint, you want to keep in mind these differences because the screening tools that you would typically use to identify IDH mutant gliomas are not going to be as functional in infratentorial tumors. And so you're just going to want to keep it in mind, keep this diagnosis in the back of your head, because these are actually easy to test for from a molecular standpoint. But if you don't remember that this entity exists, you're not going to test for it. All right, so this is a TS&E plot from a global methylation profile. I'll talk a lot more about global methylation profiling later, but the important thing here is that IDH mutant infratentorial astrocytoma does segregate as a somewhat distinct entity from the remainder of IDH mutant astrocytomas. And it also exhibits a somewhat worse survival than supratentorial counterparts, still performing better than IDH-wildty GBM, and in this particular case, diffuse midline glioma with K27M mutation. So this suggests that it actually is somewhat distinct pathogenically, although one questions whether or not this reduced survival may just have to do with the fact that it's growing in a place that really doesn't accommodate extensive tumor expansion. That really remains to be seen. Okay, enough of that. Let me move on right now to the meat of the lecture. I'm going to first talk about pediatric high-grade gliomas. And so these tumors were often suspected to be molecularly distinct from adult high-grade gliomas, even though histopathologically they were quite similar with their cellularity, microvascular proliferation, necrosis, what have you. So a very important discovery was made about 10 years ago, implicating mutations in genes that encode for histone monomers, usually H3.3 histone monomers in pediatric gliomas. And these were implicated in both of the two main flavors of pediatric high-grade glioma, the supratentorial gliomas and what we refer to now as the diffuse midline gliomas. What were then referred to as diffuse intrinsic pontine gliomas or brainstem gliomas. So these two major flavors of glioma both featured high rates of mutations in histone monomers. And where the mutations were more interesting. So let me just give you a little bit of necessary background on histone protein. So this is a histone quatrimer around which DNA is wrapped in the core component of a nucleosome, which is how DNA is wrapped in eukaryotic organisms. The H3 monomers here, and its regulatory protein tail is actually sticking out here. And it's residues on this protein tail that are mutated in these pediatric high-grade gliomas. Specifically, two of them. There's this lysine at position 27, or sort of K27, and then there's this glycine at position 34, which is mutated to either arginine or valine. So K27 is a residue that's typically involved in covalent modifications to actually regulate how the DNA underlying this nucleosome is wrapped and accessible to the transcriptional apparatus. It's a very important residue. It's acetylated, methylated, what have you, and that code actually determines the extent to which the DNA is accessible. And while the glycine in position 34 is not modified this way, it is two residues upstream of another lysine that also undergoes extensive modification, K36. And what's even more interesting about these mutations is that they have this regional segregation. The K27M mutation is entirely restricted to the midline gliomas, and the G34RB mutations are restricted to the supratentorial gliomas. And so you have two entities that have emerged from this, diffuse midline glioma, H3K27 altered, and diffuse hemispheric glioma, H3G34R mutant, both WHO grade 4 aggressive tumors, although and on the spectrum of WHO grade 4, the K27M tumors are probably worse. All right, so let's talk about each of those entities individually. So first, diffuse midline glioma, H3K27 altered CNS WHO grade 4. Just to remind you, these can arise anywhere from the basal forebrain structures all the way down to the spinal cord. And they can drift into the adult population, although they're characteristically associated with children. Morphologically, they look like a diffusely infiltrating astrocytoma. A lot of features that you would typically expect to find with those. They can have microvascular proliferation, necrosis, other high-grade features. However, those are not really those are not always identified, and this may, in fact, in part be due to the limited sampling that you get with the biopsies and with these sorts of tumors. But it may also simply have to do with the fact that you don't see that all the time. These are diffusely infiltrating lesions. They tend to kill you because they're growing and usually in areas which don't accommodate a lot of tumor growth, and can be even for something that's nominally at least low grade in its histopathological appearance. Okay, so as I mentioned before, these are now we define these by the presence of mutations involving K27 in either histone 3 or histone 3.3 or histone 3.1. And so this is one of the papers that laid out that definitively, as you see, DIPG, midline, gliomas, highly enriched in these. Interestingly enough, there's also a subgroup that has amplifications in EGFR. This is actually recognized as a subset by the WHO as well now, diffuse midline glioma, EGFR amplified. One more thing I want to point out is that particularly the H3.1 K27M mutant tumors, they tend to also be enriched in activating mutations in this gene ACVR1. This is a receptor that ends up stimulating bone morphogenic protein signaling. And interestingly enough, mutations in ACVR1 and the germline are actually associated with this unusual condition, fibrodysplasia ossificans progressiva. It's a genetic condition where patients actually form ectopic bone at sites of sometimes even mild trauma, and they can end up being encased in their own skeletons. So these are exactly the same mutations that you see in DIPG for some reason. What exactly the biology is that's being mobilized here is not entirely clear, although it has been shown in mouse models that ACVR1 mutations do collaborate with K27M mutations to induce diffuse midline glioma formation. Okay, so what does K27M mutation do? Well, it turns out that K27, the residue itself, is methylated by an enzymatic activity known as the polycomb repressive complex 2. And this tends to repress underlying chromatin. So this is summarizing a lot of work, and it is somewhat of a simplification, but basically you can think of the K27M mutation as a dominant negative sink for the PRC2. It basically grabs the enzyme and doesn't let it go. So it can't actually methylate K27 residues on mutated histones and even unmutated H3.3 histones. And so what you end up seeing is this depletion of the K27 trimethyl mark, which is the repressive mark associated with PRC2. And you actually have an immunostain for that. And what you see is this dramatic loss of staining in the tumor cells with retention of staining in tumor cell nuclei, sort of like what you would expect with an ATRX stain in an ATRX mutant tumor. Interestingly enough, very rarely there are K27I mutations in diffuse midline glioma, and those actually have the same effect. So, but above and beyond that, it's not entirely clear what this is doing. Presumably, these are reactivating certain gene sets which are not supposed to be activated, which perhaps maintain cells in stem-like states that are more amenable to self-renewal, more amenable to pathogenesis. It's not entirely clear. But from a pragmatic matter, we have screening tools now for these. So this brings me to a very important point, and the point basically boils down to don't be stupid. So it turns out there are antibodies that are directed against the K27M mutation itself, in addition to antibodies directed against K27 trimethyl. And these have, of course, diametrically opposed readouts. In a K27M mutant tumor, you expect universal expression of reactivity to K27M antibody, but you would expect loss of expression of K27M trimethyl with only retention in the vasculature. So once again, it's diametrically opposite readouts. Unfortunately, these have very similar names. And so it's not uncommon, unfortunately, for these to be confused. And it's very important for you to recognize that possibility. And when you're reviewing slides, particularly sent from an outside institution, or even reviewing reports that have been written from outside institutions. Okay. So there's one more wrinkle to the story that I want to go through because I think it reveals some very cool and interesting biology. So my friend Sriram Vanetti at the University of Michigan, he actually reported this a few years ago, which is that a specific type of posterior fossa ependymoma, what we refer to now as the posterior fossa type A ependymoma, actually has the same phenotype with regard to H3K27 trimethyl that diffuse midline gliomas have. They have this loss, this dramatic loss of staining in the tumor cells with retention in the vasculature. And this is really thought to be funny at the time because these tumors, these posterior fossa ependymomas, don't have K27M mutations, except that they actually do. A very small percentage of them do in fact have K27M mutations. But of course, they all exhibit this phenotype. So what's going on with the rest of these tumors? Well, it turns out the rest of them overexpress a protein, which we now call EZHIP, enhanced derivative zest inhibitory protein. And this protein actually has within its structure an epitope that phenocopies the K27M mutation itself. Here's the methionine residue right here. So essentially, high levels of this protein will soak up the PRC2 in a similar fashion to what K27M mutations on the histones themselves. And this has actually been demonstrated experimentally. You can take this protein and in either wild type, or interestingly, if you mutate the methionine to an isoleucine, once again, you actually deplete the extent to which this protein, the extent of K27 dimethyl and trimethylation in impacted cells. And you can do the reverse experiment where you take this methionine and you mutate it into a lysine, and then actually this protein then gets methylated by the PRC2, as you can see here. So this is an incredibly cool story that just shows the extent to which overlapping pathogenic mechanisms can influence tumorigenesis in two different cellular contexts. So in posterior fossa ependymomas, the EZHIP mechanism predominates, but it turns out in K27M or K27 altered diffuse midline gliomas, you can actually get EZHIP overexpression, a very small percentage. So you get the predominant pathogenic mechanism is the K27M mutation, but you can also get K27I mutations and EZHIP overexpression in a small subset of tumors. So that's why in the latest edition of the WHO, this is now called diffuse midline glioma H3K27 altered and not H3K27M mutant. So long story short. All right, let's move on and talk about diffuse hemispheric glioma, H3G34R mutant CNS WHO grade 4. So this exhibits histopathology characteristic of supratentorial high-grade gliomas like IDH-wildty GBM. It can be enriched, however, in this small cell architecture, which is reminiscent of PNET. These are a couple of different examples of that. So it's something to keep in mind. All right, so less is known about the pathogenic effects of the G34R mutation than the K27M mutation. The biology is just more complicated. However, a couple of recent papers have been illuminating in two respects. The first is that it turns out these tumors probably aren't technically gliomas. They're actually derived, or at least likely derived, as we can tell from gene expression signatures and mouse modeling results, from interneuron precursor cells that have a unique transcription factor signature, as you can see here, GCX, DLX12. It's not important to remember those. It's just they have the transcription factor posture of an interneuron precursor cell. And the other thing that posture does is it actually facilitates the mobilization of a bona fide oncogene, PDGFRA, the primary receptor for platelet-derived growth factor, right? And what's interesting in these tumors is that a lot of these tumors then will take advantage of that mobilization by engineering pathogenic mutations within PDGFRA. And this has actually been shown. You see here G34R mutant tumors compared to K27M mutant tumors, and the enrichment of PDGFRA mutations is obvious. This figure up here describes just the interneuron precursors and where they tend to be located. Interestingly enough, when you actually experimentally delve into this a little bit more, and you culture neocortical versus brainstem precursors, the G34R mutation is really only proliferative in the neocortical precursors, not in the brainstem precursors. So that's interesting in and of itself. You're talking about a unique pairing between a susceptible cell of origin with a specific molecular alteration that only works in that cell of origin. So the other thing I want to mention here is the very high rates of ATRX mutation that you see in G34R mutant diffuse hemispheric gliomas. And this is in contrast to the extent to which you see them in K27M mutant diffuse mutant gliomas. They are present there, but in less than half, whereas they really are a defining feature in diffuse hemispheric glioma, G34R mutant. And so to the extent that this can actually be a reasonably good screening tool to use. If this comes back negative by IHC, or at least if there's loss of nuclear expression of ATRX, this is one of the diagnostic entities you should consider, particularly in the setting of an absence of IDH mutation. Okay, and these are mutations that can be tested fairly easily by molecular testing. All right. Okay, so there are high-grade gliomas in the pediatric type distribution that do not have histone mutations. And the WHO has sort of lumped those together into one diagnostic category at this point. Probably additional diagnostic categories may emerge from this over time, but at this point, we just consider these now diffuse pediatric type, high-grade glioma, H3 wild type, and IDH wild type. So really defined by what they are not. Similar in concept, at least to adult IDH wild type GBMs, the patterns of molecular alterations are heterogeneous here. You see in recurrent involvement of established oncogenic genes like BRAF, FGFR, EGFR, MYC, what have you, but in like I said, a heterogeneous pattern. And they've been subdivided into a couple of different groups, RTK1, RTK2, MYC, and this RTK1 has this association with Lynch syndrome. And that's really all I have to say about that at this point. Like I said, more data is probably going to further clarify the diagnostic breakdown of this subcategory, except for one thing, which is these MYC amplified high-grade gliomas. You tend not to see those in the adult population, and they can have distinctive histopathological features, and particularly this biphasic pattern has been reported, where you have a more diffusely infiltrative component next to a sort of obviously cellular component, high NC ratio, almost like with PNET features. So it's just something to keep in mind. All right, and finally, there's one more pediatric high-grade glioma entity I want to discuss, and that's a newly described entity called infant-type hemispheric glioma. As I said, very recently described, in part designated once again because of a unique global DNA methylation signature, which segregates it separately from other related high-grade glioma entities within the pediatric population. In fact, it's most closely related by methylation profiling to desmoplastic infantile astrocytoma, desmoplastic infantile ganglioglioma. All right, so these entities actually, unlike a lot of the other pediatric and adult glioma entities that we've discussed already, they don't have a lot of mutations underlying and tend to be driven by defining fusion events that vary by category. There's a group that seems to be driven by NTRK fusions, a group that seems to be driven by ALK, ROS1, or MET fusions. So like I said, we're still learning a lot about these tumors. In fact, they're not WHO graded at this point because we don't really have that much information as to how they perform long term. There is data to suggest, however, as you see here, that they do perform quite well compared to other high-grade glioma variants. So that's an important thing to remember. Okay, all right. So I'm going to move on now to pediatric type diffuse low-grade gliomas. So this is a series of entities that have experienced really a lot of updates in the WHO 2021, and in particular, multiple novel disease designations have been delineated. But just some general concepts across the group to think about. The histopathology is variable. They can be astrocytic, oligonucleal, angiocentric in their appearance, and across those patterns, you can also have overlapping palettes of molecular alterations. The one commonality is they generally mobilize MAP kinase pathway signaling, as you'll see. So these are diffusely infiltrating gliomas, but they have extended survival. And this contrasts sharply with diffusely infiltrating gliomas of adults, which are essentially invariably fatal. And this classification, just to remind, is very much a work in progress and limited by the rarity of the tumors in question. So a lot of what we know about these entities was established from large molecular profiling studies like this one, which delineated the pattern of molecular alterations associated with varying histopathological patterns. So here you have in blue, this is pilocytic architecture, and red, more diffuse architecture, and yellow over here we have oligodendroglial architecture. You have the molecular alterations that are associated with them. So you see recurrent involvement of a relatively narrow group of molecular alterations, highly enriched in alterations involving FGFR1, duplications, point mutations, fusions, FGFR2 abnormalities make an appearance too, BRAF V600E mutations, and involvement of these transcription factors, MIB and MIB-L1, which increased studies have actually shown also have through a roundabout way tend to mobilize MAP kinase pathway signaling. Okay, so this is a more recent study that now is really a heroic effort. It looked at 1,000 of these tumors, and they grouped them actually by driving molecular alteration. What you can see here is that within these individual molecular alteration groups, histopathology once again can be quite variable. You can have pilocytic architecture, diffuse astrocytic architecture, oligodendroglial type architecture. So it's a complicated story, and individual what appear to be uniform disease entities are being broken off from this larger whole piece by piece through a variety of approaches. But and then this is where I really want to mention global DNA methylation profiling, because it's emerged as actually a very useful tool in designating new diagnostic entities and subclasses. So for those of you who don't know, there are all sorts of epigenetic factors that regulate the extent to which DNA is transcribed. And among the most important are actually methylation events in the DNA itself, and specifically involving CpG islands that typically reside in gene promoters. And the extent to which a CpG island is methylated can determine how accessible its adjacent gene is to the transcriptional apparatus and overall expression. Canonically, increased methylation is associated with decreased expression, although it's obviously more complicated than that. But from the simple sample of molecular signatures, you can actually, and these are commercially available now, you can develop an array that actually allows you to profile CpG islands across the genome and divine the different signatures that different tissues have. And as you might expect, different developmental lineage will have very different underlying DNA methylation signatures. And this speaks to different cells and cells of origin. And since that's a big determinant of tumor gene expression, it turns out that DNA methylation signatures are a great way to distinguish different tumor types apart. In fact, recent literature has shown that it might be a foolproof way to actually designate different brain tumors from each other. And of course, once you've established the pattern of known brain tumors in terms of DNA methylation space, you can use it to actually identify novel tumor entities as essentially clusters that are distinguished from other established known entities. All right. So, and this has been done, and I'll show you some examples of that. All right, so the first entity I want to discuss in terms of pediatric low-grade diffuse gliomas is angiocentric glioma. So these have a supratentorial localization. The patients often are characterized by intractable seizures, and once again, and like some of the other entities I'll talk about, as a unique DNA methylation signature. So this intractable seizure element I want to point out is this is a tumor that can be thought of under the heading of a long-term epilepsy-associated tumor, or a LIAT, which is another way to classify these tumors that has been developed over time. All right, so angiocentric gliomas, as the name would imply, they have this characteristic histopathological architecture, which focuses on an angiocentric distribution of the tumor cells. And particularly the tumor cells, they tend to be bipolar, and they tend to stretch out kind of in a radial pattern around the blood vessels with which they interact. Although there can be cellular areas too, where they can adopt sort of more fusiform fascicular architecture. This perivascular distribution is reminiscent of ependymal differentiation. And there are other signs that these tumors actually might be related to ependymomas. In particular, they tend to have this dot and ring-like positivity for EMA, which is something that you characteristically see with ependymomas. Unlike a lot of other diffuse gliomas, these are also typically stain negative for OLIG2. Mitotic rates are usually very low. These are indolent neoplasms consistent with their WHO grade 1 classification. One of the more interesting things about diffuse is they tend to be defined by a single molecular alteration involving the transcription factor MIB, in particular fusion events with the QKI gene. So these MIB QKI fusions seem to be largely restricted to angiocentric gliomas. And what this fusion does is essentially, it's been shown it actually increases expression of MIB, and of course tickles all of its downstream effectors. Okay. So contrast that now with another recently developed entity, the diffuse astrocytoma MIB or MIB-L1 altered CNS WHO grade 1. So this entity actually emerges from a diagnostic entity that was previously referred to as isomorphic diffuse glioma or isomorphic diffuse astrocytoma, so called because of this very characteristic monomorphism. These are very that tumor cells would exhibit. Paucicellular tumor, and these monomorphic, almost sort of pseudo-protoplasmic astrocytes distributed in a vaguely microcystic or background, as you can see here. So these tumors also are supertentorially located for the most part. Patients have intractable seizures. This is another LIAT. And these have a unique methylation signature, as you can see here. This is a TS&E plot that essentially allows you to cluster tumors by methylation signature. And here you can see the what's called the isomorphic diffuse glioma, segregating from most closely the angiocentric glioma. And speaking to this relationship, this tumor has alterations in MIB as well, and also another transcription factor related to MIB, MIB-L1. And these alterations increase expression of MIB and MIB-L1 as you would expect. But what's interesting about these is, particularly the MIB alterations, they do not involve QKI. The MIB QKI alterations are entirely restricted to angiocentric glioma. These MIB alterations involve other partners. So once again, it speaks to slightly different pathogenic mechanisms and slightly different cells of origin. All right. Okay. So let's talk about another entity, another supertentorial entity associated with intractable seizures falling under the LIAT subgroup with a unique DNA methylation signature. This is the polymorphous low-grade neuroepithelial tumor of the young. If you don't want to say all that, you can just say PLNTY or plenty. So this actually has variable histopathology. It can be prominently oligodendroglial, as you see here with the characteristic perinuclear halos, microcalcifications, chicken wire vasculature. However, there can be areas that are much more astrocytic in their morphology and even fusiform and spindled cell in their arrangement. There can also be areas that are reminiscent of ependymoma with perivascular pseudo-rosettes. Okay, so unlike the previous two entities I discussed, this entity tends to be positive for OLIG2, but more interestingly than OLIG2, these tend to widely and strongly express CD34 within their tumor cell population, which is an unusual feature. So unlike the two previous entities, this tumor is a little more heterogeneous in terms of its spectrum of molecular drivers. However, most of the time you're talking about BRFE600E or fusion events in FGFR2 or FGFR3. There are some NTRK fusions that have been more recently described, but really these are the more common molecular alterations that you're going to be looking at. I want to point out one thing here in particular. This FGFR3 TAC3 fusion has actually been associated with a subset of IDH-wildty glioblastomas, and it's exactly the same molecular alteration. So it's very important to recognize that if one of these pops up, you should consider the possibility that you might actually be dealing with a high-grade glioma, particularly in the context of obvious high-grade features or molecular alterations like CDKN2A, TERT mutation, what have you, that would tip you off that you're dealing with a high-grade glioma. Obviously, that's going to perform very differently from a nominally CNS WHO grade 1 tumor. Okay. All right. So I want to, let me see, is this the last entity I talk about here? Yeah. Okay, so there's one more entity I want to talk about within the pediatric low-grade diffuse glioma group, and so similar to what the WHO did with high-grade gliomas, the rest of the low-grade gliomas don't fit into the categories that I've described up until now. They've thrown into a single category called diffuse low-grade glioma MAP kinase pathway altered. So these have a broader localization pattern throughout the neuraxis. They tend to have both. They can have both astrocytic and oligodendroglial morphological features. Unlike the entities before, there is no unifying DNA methylation cluster, and there's a common but not invariable association with epilepsy. So all of this suggests to me that this is a more heterogeneous group of tumors, that likely additional distinct diagnostic entities will be broken off here in the years to come. Once again, we're talking about a fairly heterogeneous spectrum of molecular alterations that define it. However, some are more common than others. In particular, FGFR1 duplications and point mutations and BRAF V600E mutations predominate over others like NTRK mutations, MAP2K1 mutations, MET abnormalities, and what have you. It's important that these are really defined by what they are not. These are IDH-wildty, H3.3 wild type, and there is no CDKN2A loss, which suggests actually a higher grade tumor. And so indolent behavior is the rule for these, but there is no formal WHO grading as of yet, likely due to the heterogeneity within the subclass that I've mentioned already. Okay, all right. So let me see how am I doing my time. Okay, so the last thing I want to talk about are the circumscribed astrocytic gliomas, and the WHO recognize 6 of these at this point, but I'm only really going to talk about 3 of them. I'm not going to talk about polycytic astrocytoma and pleomorphic xanthoastrocytoma because these are fairly common, and I figure you guys know about these already. I'm also not going to talk about SEGA because the pathogenic mechanism that is pretty well defined in association with TSC. So I'm going to focus on these other 3. Okay, so the first is choroidoid glioma CNS WHO grade 2. So this sort of epitomizes the notion of a circumscribed astrocytic glioma. These were often before called choroidoid gliomas of the third ventricle because of this very characteristic location within the third ventricle. And this is thought to do with the fact that they arise from, their thoughts arise at least from specialized tanocytic ependymal cells within the organum vasculosum and lamina terminalis. And because there's this sort of unique localization pattern, they tend to rise with symptoms of obstructive hydrocephalus, compression of the hypothalamus, and optic chiasm. They have this very characteristic choroidoid architecture with epithelioid cords of cells immersed in this myxoid matrix. They also tend to be very well demarcated. Here you can see the surrounding brain infused with Rosenthal fibers, sort of characteristic of the brain trying to wall off a disease process. Okay, so akin to other tumors arising in the vague distribution of the hypothalamus, these tend to be TTF1 positive. They also are CD34 expressing avidly. But so an interesting story has emerged recently on choroidoid gliomas, in that they essentially are now associated with a defining mutation involving protein kinase C, the alpha subunit. So here's the paper that described it. Interestingly enough, they were able to actually put this mutation, a mutated form of this, into astrocytic precursors and demonstrate oncogenicity. And so this is, I think, a great example of a unique cell of origin coupled with a unique molecular driver that then results in a completely unique tumor. Okay. All right, so let's talk about astroblastoma next. To me, this epitomizes the notion, or in fact, the tension between an architectural pattern and an actual disease entity. So astroblastoma is, in fact, initially described as an architectural pattern. Astroblastoma is architecture, which vaguely resembles ependymoma in these perivascular pseudorosette development. The difference is that the processes of astroblastoma is that they're directed towards the center of these vessels, are usually broad-based, sometimes referred to as fat processes. Interestingly enough, these pseudorosette structures can become extensively hyalinized to the point where you get this pseudopapillary architecture with these extensively hyalinized perivascular nodules. This relationship with ependymoma is emphasized by the fact that a lot of these tumors exhibit positive immunostaining, either in a dot and ring-like or even membranous type distribution for EMA. And you can also get this more confluent peanut type architecture in these tumors, which is actually important because of I'll get to why it is in terms of how they were initially designated. Something else I want to mention is that these have a female predominance, which is unusual among brain tumors. So you might want to keep that in mind. Okay, so this gets back to this notion of peanut architecture in astroblastomas and the contribution it made to how they were originally designated, at least this particular MN1 mutated or MN1 altered diagnostic entity. So this was a paper from a few years ago now, which actually looked at NDs broadly described as CNS supratentorial peanut. And what they found by, once again, methylation profiling is that most of these tumors actually belong to other diagnostic categories, and most specifically high-grade glioma and its various forms. However, 3 small entities did actually emerge from this, one of which was referred to as CNS high-grade neuroepithelial tumor with MN1 alterations. And it turned out that tumor set was enriched in astroblastomatous-like architectures you can see from these pseudopapillary forms. So since then, other groups have gone back and actually looked at astroblastoma, tumors described as astroblastomas in particular, and they've identified subsets which actually had these fusion events involving MN1. And those tumors were molecularly distinct from the tumors that did not have MN1 fusions. And in fact, the tumors in the latter group tended to have molecular alterations that you typically associate with, IDH-wild type high-grade glioma. So it's important you actually distinguish these because although MN1 fused astroblastomas are still being studied in terms of their prognostic features and whatnot, so they're not officially WHO graded at this point, there's a general sense that they do perform more favorably than IDH-wild type glioblastoma. So it's important that you recognize this distinction. All right, so finally, I'm going to talk about high-grade astrocytoma with pyloid features, yet another novel entity to emerge in the new WHO 2021 classification. So before we have these notions of anaplastic pilocytic astrocytoma, I don't want you to think that that's what this is. There is a predilection for the posterior fossa in these tumors. However, the median age of these tumors is older, and importantly, most of these tumors actually arise de novo, and they don't emerge from previously known low-grade pilocytic astrocytomas. So there's this feeling that this might actually be a distinct disease entity. So you can get Rosenthal fibers and eosinophilic granular bodies, but not necessarily in the majority of tumors, more like a third. The histopathology itself can be quite variable. You can get tumors that look, frankly, like IDH-wild type GBMs, like PXAs. There can be enrichment in pyloid architecture. Most have molecular microvascular proliferation, as you can see here, and about a third will have necrosis. So this is really the landmark paper that established this diagnostic entity. As you can see, once again, there's a unique DNA methylation signature that separates it from other pilocytic astrocytoma subtypes. Clinically, these perform worse than other pilocytic astrocytomas. In fact, their survival tends to resemble IDH-mutant WHO grade 4 astrocytomas, what we used to refer to as IDH-mutant glioblastoma. So not exactly a benign disease here, but still better than an IDH-wild type glioblastoma. And once again, enrichment of MAP kinase pathway alterations in terms of its driving molecular alterations. You see a prominent representation of the KIA1549 BRAF fusion protein, which you see in pilocytic astrocytomas, but there are also a lot of NF1 mutations, FGFR1 mutations and fusions, even KRAS mutations. Interestingly, BRAF V600E is not very well represented in this entity, so that's an important characteristic. And finally, as a unifying feature, CDKN2A deletion is very common in these tumors, as is ATRX mutation. So those are also good things to remember if you're considering this diagnostically. All right, so I've gone through a lot of stuff today. I just want to briefly review some of the major themes. So the first, IDH-mutant astrocytomas can arise infratentorially with sort of unique molecular features. Pediatric-type high-grade gliomas are defined by epigenetic abnormalities, specifically mutations in H3.3 and H3.1 histone monomers. Pediatric-type low-grade gliomas feature MAP kinase pathway activation. Pediatric-type low-grade glioma subclasses have emerged with the aid of integrated molecular profiling, including global DNA methylation profiling analysis. So discrete molecular alterations characterize subsets of circumscribed astrocytic gliomas. We talked about the protein kinase C mutation and choroid glioma. We talked about the MN1 alterations in astroblastoma. And finally, a unique DNA methylation signature defines high-grade astrocytoma with pilocytic features. Okay, so just to remind you, we did this case, 4-year-old with a history of intractable seizures, these histopathological features, this molecular characteristic, this is a plenty. All right. So I just want to thank Greg Fuller and Leo Baster, my partners here, who provided some of the images that you saw today. And I'm happy to take questions. Thanks so much for your attention.
Thank you very much, Dr. Huse. At this time, we'll move to the Q&A. You may submit your questions via the chat box or unmute to ask a question. When submitting a question via the chat box, please ensure the message is to everyone so that the presenter and attendees are able to see the question. To unmute, please select the microphone in the lower left corner. Once you've asked your question, please remute to avoid background noise. So Jason, it looks like we are already seeing some questions in the chat. Thank you so much. That was a fantastic trip through a lot of information. So we really appreciate it. So if you want to start looking at the questions, you can take it away. If not, I'm happy to read them.
Okay, so I can start from the top here. There's a question from Mohammed Harry. He says, in PLNT, is FGFR3 TAC3 methylation profiling clustered with BRAF mutation ones? Yes, at least from what we can tell right now, they appear to overlap in their clustering. All right, so moving down to one from Namita Sinha. How do you differentiate EGFR amplified and TERT promoter mutated IDH wild type RTK2 pediatric high-grade glioma from adult glioblastoma? That's a great question. It's difficult, and in fact, they really may be the same entity when it really comes down to it. I mean, this is what we're starting to see, is that these diseases that are characteristically associated with one population, or we see them drifting into other populations non-canonically. We are in fact human beings after all, even the little ones. So that's my feeling on that, although that may not be an entirely satisfying answer. All right, so question from Francisco Garagori. In diffuse midline glioma, H3K27 altered, loss of H3K27 trimethyl is stated as essential criteria. Does it imply that using H3K27 trimethyl could be used as a screening tool for to discard the possibility of this type of glioma? Yes. In fact, what we typically do is we run both K27 trimethyl and K27M, but K27 trimethyl is valuable because, like I said, there are other tumors like posterior fossa ependymoma that actually sort of have an overlapping distribution in terms of geography that you may want to consider. That are also H3K27 trimethyl loss. It's just definitely get your immunolab to make the names of the antibody stains as distinctive as possible, so that it reduces the likelihood of them being confused with each other. I can't stress that enough. Okay, so I want a question from Melissa Blessing. Hi, Melissa. Do you do CD34 on most low-grade gliomas, given the polymorphism? Yes, actually, particularly, low-grade gliomas, I will actually throw that on there. Sometimes because it's hard to detect whether they're really infiltrative or whether they're encapsulated, and particularly with an entity like with a lot of these entities that are more solely that are indolent, you can often also see the ramified neuroepithelial elements on the periphery of the tumors that can help you actually make a diagnosis of like ganglioglioma, for instance, or whatnot. So I find CD34 to be an incredibly valuable stain. Okay, so from Beatrice Lopez, hi Bea. When should we suspect of high-grade glioma with pyloid features? Yeah, no, it's something that you really have to consider. It's got variable architecture. It can diagnostically overlap with standard GBMs for sure, although they tend to be localized more prominently in the posterior fossa. But this is one of these situations where I think obviously if you see some architectural features that maybe tip you off. The other thing that I think is very valuable, and I tell people this all the time, and it's not just because I study this protein for a living, but ATRX immunostaining is really valuable. I think because it not only designates IDH wild type or IDH mutant astrocytomas, there's a whole subclass of tumors where you see ATRX deficiency it really increases the likelihood that you're dealing with one of those entities when you actually see it. So I would encourage that. I hope that's a satisfying answer. Okay, so another question from Francisco Garagori. How do you distinguish between diffuse pediatric type high-grade glioma H3 wild type and IDH wild type RTK with EGFR amplification? Okay, so I think this overlaps with the question that was asked earlier, but yeah, it's tough. And frankly, I'm not sure if you really can distinguish with them. I think they may actually be ultimately the same entity when it all comes down to it. I don't know. It's very tricky. They certainly it's something that would be definitely worth studying from the standpoint of global methylation profiling. For instance, if anybody has a large collection, I mean, I know everybody has a large collection of EGFR amplified IDH wild type adult gliomas, but the question is the EGFR mutant diffuse midline gliomas, the EGFR mutant pediatric type high grade gliomas. It would be very interesting to see how they segregate out. Okay, so a question for Josh Kolonsky. What is your group's approach to P16 and CDKN2A? So this is, I will answer that question saying my group's approach is suboptimal because we're doing FISH still. Hopefully soon, when we transfer over to global methylation profiling as a clinical tool, we'll be able to do it more effectively on the methylation platform, because then we'll get good tiling through that entire region. But now we're using FISH, and it can be tricky to interpret, particularly because our cytotechnologists, they're not used to looking at CDKN2A in gliomas, and we just sort of sprang this on them over the last year and a half. But we tend to do it on first of all on any IDH mutant astrocytoma, because it's now required or a required element, and can actually in and of itself cause upgrading of something to WHO grade 4 when it's present, when homozygous deletion is present. But like I said, it's present in a lot of the other entities, and I think I tend to use it as liberal as I can if it offers some assistance in terms of diagnosis. One of the issues though is that while CDKN2A it helps to distinguish lower grade glioma, the higher grade tumors that I talked about here from other lower grade gliomas, unfortunately CDKN2A loss doesn't do a good job in distinguishing these entities from like high grade gliomas. Like for instance, CDKN2A is one of the most prominent losses, one of the most prominent molecular alterations that you see in IDH wild type glioblastoma, for instance. So if you're trying to distinguish something from IDH wild type glioblastoma, it's probably not the best approach to use. Okay. I think I got through everything there. Oh, whoops. Here we go. From Carrie. Hi, Carrie Mahila. Your original PLNT paper left room for those categorized as neuroepithelial tumors. Yeah. Have you been finding many PLNT that have a neuronal component, sometimes prominent? The new WHO has grouped these as diffuse LDGs. Can you comment on the categorization? Still a work in progress. I think the short answer is yes. I mean, and that's not, and I'll make it even more of a cop out by saying that could be true of actually a lot of the entities that we talked about today. For instance, the diffuse hemispheric gliomas, are we going to start referring to those as neuroblastomas or something? I don't know. I mean, I think yeah, the molecular markers expressed in these can be quite variable, and they're reflective ultimately of the fact that a lot of these are, I think, derived from multipotent neuroepithelial precursors, and their differentiation is really kind of a gamish of haphazardly reactivated developmental programs, what have you. So it's tough, but I could definitely see plenty flopping back and forth, and eventually I could see these entities I talked about today in terms of low-grade diffuse gliomas being merged with some of the related tumors in the glioneuronal group as to kind of a neuroepithelial, low-grade neuroepithelial group. It could definitely happen. Yeah, like I said, another completely unsatisfying answer. All right, so now question from Namita Singh. I worry about misdiagnosing H-gap from a pilocytic astrocytoma when the posterior fossa tumor has Rosenthal material and EGBs and mutation present. Do you recommend using CDKN2A codel studies in these posterior? Yes, I think that's a great idea, particularly if the tumor is high grade histopathologically. Yeah. Okay. So a question from Romain Carriol. What is the value of OLIG2 and P53 ATRX loss H3 G34 mutant glioma? Well, I mean, to be perfectly honest, we tend not to use OLIG2 in that situation. P53 and ATRX loss, P53 mutation, so positive P53 and ATRX loss, yes, definitely. That's very important. In fact, that's how we usually pick them up. We'll get a high grade glioma in a young adult, because I mean, I work at MD Anderson, so we don't get too many kids here. So it's like a 30-something with a high grade glioma. It'll be ATRX deficient, but will not have IDH mutation. That's usually the tip off. Then we know when we send that thing for sequencing, we know it's going to come back with a G34R mutation or G34B mutation. So yeah, so I would definitely use those things.
Right. Looks like that's the end of it. Maybe, I mean, we have 4 more minutes. I do have about the CDKN2A loss and associated with BRAF mutations. I always get stuck with this, like the definition of, pediatric-type low-grade gliomas, MAPK altered. They have to have absence of CDKN2A loss, right? So then what happens if you have a diffuse low-grade glioma with BRAF and then you run into CDKN2A loss? What do you call that? What do you do with that?
Well, so it depends on the situation, but there are a couple diagnostic entities you consider. The first is high-grade astrocytoma with polycytic features. The second would be PXA, in particular anaplastic PXA, exactly. Which is something that also tends to fall into that category. So it's and the third is even GBM. So I think it still remains to be seen. I'm not one. The extent to which BRAF V600E in and of itself relegates an otherwise high-grade glioma into the PXA category. Certainly, there's a large subset of what I could refer to now as epithelioid GBMs with BRAF V600E mutations that are anaplastic PXAs, but I'm not sure if they all are yet. I think it really remains for the field to sort that out. Yeah. Right. Yeah, and in that context, CDKN2A loss is not necessarily a high-grade feature, right? Like in the PXA world, the coexistence of those two things does not mean it's a higher grade. Yeah, I mean, I think they are. It doesn't necessarily mean it's a high-grade PXA, but they are. The papers on anaplastic PXA have pointed to CDKN2A loss as a prominent recurrent feature. Yeah. There was somebody else asking a question, I think. Yeah, Dr. Santi, were you saying something?
I think that, all you said is correct, but the morphology has to be considered. I mean, a PXA has to have the feature of the PXA. Sure. I mean, the high grade, now there is this high grade pilocytic astrocytoma with the pyloid feature, but you have to put everything together. GBM with BRAF normally as a giant cell GBM, but has to be a GBM. The morphology still counts. If we use exclusively the molecular, we will have a lot of problems. Yeah,
we still have a job. Yeah, clinging to a job by our fingernails. We still have a job.
All right. Well, thank you so much, Jason. That was fantastic.
Yeah, thanks so much for tuning in. I really enjoyed this.
Yep. Thank you again for joining today's AANP Teaching Rounds presentation. We would ask that you take a few minutes to complete a short evaluation. Completion helps ensure accurate reporting to the accreditation board. A link to the evaluation will appear on your Path LMS screen upon the conclusion of the Zoom meeting, or you can navigate back to the main course and select the evaluation option. Once you have completed the evaluation and selected your credit amount, please then select the appropriate certificate based on your credentials. The PowerPoint slides and recording will be posted to the AANP website in the next week. Thank you again, Dr. Huse, for an excellent presentation. This concludes the session for today.